Good evening, dear students. Can you see the screen? Okay. So today we are going to start with the next chapter, that is biomolecules. Okay. Very simple chapter. So here we are going to discuss about the MCQs. Okay. Already we have seen the synopsis. Okay. So let us solve. So this is the questions on the biomolecules. Okay, so we know the important biomolecules. Okay, carbohydrates, proteins, vitamins, right? Nucleic acids, right? So on these particular biomolecules, okay, so let us solve some of the MCQs. Okay. Yeah. So this is the first question. The number of disulfide linkage present in insulin are. What is the number of disulfide linkage present in insulin? What is the number of disulfide linkage present in insulin? What is insulin? What is insulin? Yes. Disulfide. Disulfide linkage present in insulin. Yes. What is the answer? Okay. Sanjeevni has answered as 2. So when you see the structure of insulin, okay, when you see the structure of insulin, okay, it consists of 3 disulfide linkages. Okay, it consists of 3 disulfide linkages. So there are 2 interchain. Okay, there are 2 interchain. Interchain means what? Between different molecules. Okay, there are 2 interchain disulfide bonds. Okay, which is present between A and B chain. Between A and B, there are 2 interchain disulfide linkages. Okay, that is from A7 to B7 and A20 to B19. So these are the 2 interchain disulfide linkage, okay, which is present in insulin. And there is one intrachain. Intra means what? Within the molecule, okay, within the same. So within the same molecule, there is a chain between A6 and A11. So this is another disulfide linkage. So there are 3. So the number of disulfide linkages present are 3. Okay, so option number A is the answer. Option number A is the answer. I hope it is clear. So when you see the structure of insulin, okay, when you see the structure of insulin, okay, so when you see insulin structure, there are around 51 amino acids. Okay, so insulin, it consists of 51 amino acid and they are arranged in 2 polypeptide chains. Okay, they are arranged in 2 different polypeptide chains. So these polypeptide chain, they contain 21 and 20 amino acids. Okay, in 2 polypeptide chain, there are 21 in one and another 30 polypeptide, that is amino acid, are present. Okay, and how these two polypeptides are joined? These two polypeptides are joined together by the disulfide linkages. So they have asked how many disulfide linkages are present between in the insulin. There are three disulfide linkages are present. So there are two interchain. Interchain means between two polypeptide chain. There are two, and within the same polypeptide there is one. Okay, so this is the answer. So option number A is the answer. I hope it is clear. Yes, Lubna, is it clear? Okay, I will explain it again. Okay, so insulin. Okay, when you see the structure of insulin, so it consists of 15 amino acid, 51 amino acid. Okay, how these 51 amino acids are arranged? They are arranged in two polypeptide chain. Okay, one polypeptide chain consists of 21 amino acid, another polypeptide chain consists of 30 amino acid. Now these polypeptide chain, okay, they are joined together. How they are joined together? They are joined together by a disulfide linkage. Okay, they are joined together by disulfide linkage. So they have asked how many disulfide linkages are present in this insulin. So between inter college, inter college competition means between two different colleges. So now there are how many this so between, so this is one polypeptide chain and this is another polypeptide chain. So to hold these two polypeptide chains, so between this A and this is B. So there are two disulfide linkage. Okay, two interchain disulfide linkage are present. That is at A7 to B7. So this is one. Another is A20 to B19. So these are the two chain disulfide linkage. Another is within the same polypeptide chain, that is at A. Within this only, there is another this what disulfide linkage, that is between A6 to A11. So these are the what the number of disulfide linkage present in the insulin. Is it clear now? So because it has A has 21, because the A has 21 amino acid, within the same only it is having one more intra disulfide linkage. Okay. So internal disulfide linkage it is called. So there are total three. There are total three disulfide linkages. Okay. Let us go for the next one. A diabetic person carries a packet of glucose with him always. Why he will carry a packet of glucose always with him? What is the reason? When you see a diabetic patient, okay, when you see a diabetic patient, he always carries a packet of glucose with him. Why? What is the reason? Why they carry? Okay, Sri Varuni has answered as C. Others? Why a diabetic person carries a packet of glucose with him always? Yes, Anil Kumar has answered as B. Baby and Sanjeevni has answered as C. Okay. So why the diabetic patient, okay, or person, they carry a glucose packet, okay, so that so diabetic patient, okay, when they suppose they if they are walking or if they are doing okay, suddenly they are sure this glucose level will decrease. They may faint. So sometimes they faint. Okay, there are chances of a diabetic person so that they can faint. So why they faint? Why they feel this why they want to faint like that? Because there will be drop of blood sugar. Okay, there will be blood sugar will decrease. So if they take glucose, okay, when they take glucose, so quickly their blood sugar level will increase. Okay, if they take the glucose, what will happen? Their blood sugar level will increase immediately. Instantly, it will increase. So it will come to normal. Okay. So because of that, always a diabetic person, they carry a glucose because to increase it, the glucose, what it will do, it will increase the blood sugar level. Okay. Very fast. So very fast, it will increase their blood sugar level. Very good. So Shri Varuni, Baby and Sanjini have answered it correct. Very good. Okay. Let us go for the next one. There are 20 naturally occurring amino acids. The maximum number of tripeptides that can be obtained is. So there are 20 naturally occurring amino acids. Okay. So the maximum number of tripeptides, how much can be obtained? So tripeptides means, what is a tripeptide? What is a tripeptide? Tripeptides. So there are 20 naturally occurring amino acids. How many tripeptides can be formed? So what is a tripeptide? When three amino acids, okay, when they are joined by two or more this, okay, so three amino acids, when they are joined by the peptide bonds, okay, so then they are called as tripeptides. So how many? So you can do 20 raised to 3. So because tripeptide means they are made up of 3. So there are 20 naturally occurring amino acids. So 20 raised to 3 is how much? Cube root of 20. So 20 into 20 is 400. Again, 400 into 20 is 8,000. So it is 8,000. So the maximum number of tripeptides which can be obtained is 8,000. Okay. So option number D. Yes, Amulya, Sanjeevni, Sri Varuni has answered it correct. It is D. Lubna has answered it correct. Very good. So when there are 20 naturally occurring amino acids, so maximum number of tripeptides. Tripeptide means it is made up of 3 amino acids. So 20 raised to 3, it will be how much? It will be 8,000. Okay, very good. Fine. Let us go for the next one. The letter D in D-glucose signifies. So we write the sugars, right? Like D-glucose, D-glyceraldehyde. So what this D refers to? What does this D refers to? Yes, what it refers to? D means what? D in the D glucose. What is, so when we select the compound, okay, we know that glyceraldehyde is taken as the standard. Okay, so when you see the standard configuration, okay, of glyceraldehyde, so why D is used? It is used to it will tell you what it signifies, the configuration of a particular chiral carbon. So what it is, configuration at a particular chiral carbon, right? So in when you study the configuration in the sugar, okay, of this different sugars, so D and L, what it signifies the configuration, okay, of a particular chiral atoms. Yes, it is D. Answer is D. Okay, so D in the D glucose, what it signifies? It signifies the configuration of a particular chiral carbon. Not only D, your L, D and L forms. Okay, when you are studying the sugar chemistry, so what it signifies? It signifies the chirality. Okay, particular chiral carbon. Okay, yes. See here, when we are discussing the sugar chemistry, okay, so these are different sugars, glucose, lactose, fructose, okay. So when you are, what DL it refers to. So when we are taking sugars, glyceraldehyde is the standard sugar, okay, that is we take the standard configuration of glyceraldehyde, okay, when we study the different sugars. Now D and L. So D and L form, what it tells you? It tells you the configuration, okay, how the chiral carbon is placed, okay, what is the configuration of a particular chiral carbon. So it tells you about the configuration. So it is option number D. Okay, next question. Which one of the following does not correctly match with each other? Silk is a, which will not match with each other. A, silk is a polyamide, lipase, enzyme, butter, fat, oxytocin, enzyme. Which of the following does not correctly match with each other? Yes. Which of the following does not correctly match with each other? So silk, yes, silk is a polyamide. Okay. Silk is a polyamide. So yes, this is correct. It is correctly matched. Lipase is an enzyme. This is also correct. Butter and fat. Yes, butter is a fat. Then oxytocin. Oxytocin is not an enzyme. It is a hormone. Okay, it is a hormone. So it is a polypeptide hormone. So this is wrong. Oxytocin is a hormone. Okay. So yes, option number D is the incorrectly match. Okay. They are not correctly matched with each other. Okay. So option number D is your answer. Fine. Let us go for the next one. One of the following is an essential amino acids. Which of the following is an essential amino acids? Which of the following is a essential amino acids? Yes. A cysteine, B serine, C tyrosine, and D isoleucine. What is an essential amino acid? Essential amino acid. So essential amino acids are what? They are not essential amino acids are not prepared in our body. They are to be taken through diet, right? So which is the example? It is D. Okay. So isoleucine, it is a essential amino acid. Okay, it should not, it is not prepared naturally, it is not obtained naturally, it should be taken through diet. Okay, that is essential amino acid, isoleucine. Next is glycogen is, what is a glycogen? Glycogen is a structural polysaccharide, structurally similar to amylopectin, but extensively. C, a polymer of beta D glucose unit. And D, structurally very much similar to amylopeptine. What is glycogen? Only branched. Okay, structurally similar to amylopeptine but extensively branched. Yes, structurally similar to amylopeptine but extensively branched. Okay, Sri Varuni has answered it as B. What is glycogen? What it is? It is the animal starch, right? Glycogen is what? It is the animal starch, right? And how is the structure? It is structure is similar to that of the amylopeptine, right? It is the structure is between the amylopeptine. But what is the difference between the glycogen and amylopeptine? In amylopeptine, okay, uh, it is a huge molecule. Okay, the structure is huge. It is made up of 20 to 25 glucose units. Okay, and in glycogen, it is very small or it is very short. It consists of 10 to 14 glucose unit. Okay, so glycogen is highly branched. So the answer is what? Answer is B. Structurally similar to amylopeptine, but it is highly branched. So yes, option number B is the correct answer. Option number B is the correct answer. Okay. So what is glycogen? So glycogen, as we know, it is the animal starch. Okay. It is the animal starch and the structure. Okay. When you see the amylopeptine, yes, glycogen and amylopeptine, they are having similar structure. But amylopeptine, it is having a little larger structure. It is made up of 20 to 25 glucose unit. And in glycogen, it is made up of 10 to 14 units of glucose. But glycogen is highly branched. Branching is more, okay, in glycogen. So option number B is the answer. Okay. Let us go for the next one. Which of the following is correct about hydrogen bonding in DNA? So, which of the following is correct about hydrogen bonding in DNA? Yes, go through the options and tell me. So, this is about your nucleic acid, right? When you see the bonding of nucleic acid, so you have four: adenine, thymine, guanine, and cytosine. So, how the pairing will take place? So, in DNA, when you see hydrogen bonding, adenine, okay, it forms two hydrogen bond with the thymine, right? And your guanine, it forms what? It forms three hydrogen bonding with the cytosine. So, which is correct? Option number A, right? A and T and G and C. So the correct hydrogen bonding is A will pair up with T with two hydrogen bonding and the guanine will pair up with C with three uh, bonding. Okay, three hydrogen bonds. Okay, fine. Yes. See here in DNA, okay, when the DNA sequence, you know, in DNA there are four bases, right? In DNA there are four bases, that is adenine, then guanine, okay, thymine and cytosine. So when these bases are paired, okay, when you are writing the DNA structure, so how these bases are paired, how they are paired? Adenine is paired with thymine, okay, with two hydrogen bonding, and your guanine is paired with cytosine with three hydrogen bonding. So like this is the pair. Okay, you cannot pair A and C. This is only the pair which is present. Okay, but in RNA, instead of thymine, what is present? Your uracil is present. Okay, this is the only change in RNA. So option number A is the answer. So this is the pairing taking place. Next is hormones are secreted by ductless glands of human body. Iodine containing hormone is. Hormones are secreted by ductless glands of human body. Iodine containing hormone is A. Insulin, B. Adrenaline, C. Testosterone, and D. Thyroxine. Yes, hormones, how they are secreted? They are secreted by a ductless glands. Okay, so which is the hormone which contains iodine? Yes, yes. Karina, Shrivaruni and Sanjeevni has answered correct. It is D, thyroxine. So thyroxine, it is the what? It is the hormone which consists of iodine. Okay, so thyroxine, very good. Okay. Next question. Which of the following is generally water insoluble? Which of the following is generally water insoluble? A. Fibrous protein. B. Vitamin C. C. Amylase, and D. Glycine. Which of the following is water insoluble? Yes. Which of the following is water insoluble? So it is fibrous protein. Okay, it is fibrous protein. So fibrous proteins, they are water insoluble. The rest all, they are water soluble. Okay. So when we see the fibrous protein, they are water insoluble. Vitamin C, amylase, and glycine, these are all water soluble. Okay. Next is C1 to C4 glycosidic bond is not found in A. Sucrose, B. Lactose, C. Starch, and D. Maltose. C1 to C4 glycosidic bond is not found in. Yes, it is not found in what? It is not found in sucrose, because in sucrose you have C1 to C2 glycosidic linkage. Okay, when you, so for this you need to see the structures. Okay, all the structures of the sugars. Okay, glucose, lactose, starch, maltose, all these sugars you need to see the structures. Okay, so the glycosidic linkage between in the sucrose is C1 to C2, and rest all lactose, starch, and maltose, it consists of C1 to C4 glycosidic linkage. So sucrose is the sugar. Okay, it does not consist of C1 to C4 glycosidic bond because it consists of C1 to C2. Okay, fine. Next, RNA and DNA are chiral molecules. Their chirality is due to the presence of RNA and DNA. They are chiral molecules. Their chirality is due to the presence of. Why? Yes, tell me the answer. What is this? Oxygen is present. This is your beta D ribose. Okay, then HOH, CH2OH, and H. So here there is no oxygen. So this is your beta D2 deoxyribose. Right. Now here, why DNA and RNA, they are chiral molecules, right? Their chirality is due to the presence of. So what you can see here, in RNA and DNA, okay, they have ribose and deoxyribose sugar. In RNA, what you have? In RNA, you have ribose sugar, and in DNA, you have deoxyribose sugar. Okay, so they are chiral. Okay, both these are chiral compound. Okay, why? Because of the presence of this D-ribose and D2 deoxyribose sugar. Okay, in DNA, what you can see, the in DNA, what is the sugar moiety? it is beta D 2 deoxyribose. Okay. And in RNA, which is the sugar, it is beta D ribose sugar. Right. So here it is due to what? D sugar component. So you can see the sugar component is D. Okay. So DNA and RNA are chiral molecules. The chirality it is due to the presence of this D sugars. Okay. Uh, because of this D sugar component, okay, their chirality is C. So option number A. Option number A. Okay. So DNA and RNA, they are both chiral. Okay. You can see they consist of ribose and deoxyribose sugar. So what is the main component here? The D sugar component. So because of that, you can see the chirality. Okay. Next. In nucleic acid, the molecules are joined together by. So in nucleic acid, how the molecules are joined together? By phosphoester linkage, phosphodiester linkage, phosphodisulfide linkage, or sulfodiester linkage. Very important. In nucleic acid, how is the linkage? Yes. How is the linkage? It is by phosphodiester linkage. Okay. So in nucleic acid, the molecules are joined together by phosphodiester linkage. Remember that, don't forget, very important. So in nucleic acid, how the molecules are joined? What is the issue? How it is joined together? They are joined together by a phosphodiester linkage. Okay, fine. Next question. The two forms of D-glucopyranose are called as. So when you have two forms of D-glucopyranose, they are called as what? Diastereomers, anomers, epimers of enantiomers. Yes, the two forms of D-glucopyranose, what they are called as? Yes, the two forms of D-glucopyranose, they are called as what? They are called as anomers. Okay, they are called as anomers. Okay, anomers. Okay. What are anomers? Anybody? What are anomers? Anomers. What are anomers? Yes, we. So, what the question was asked, the two forms of D-glucopyranose, they are called as what? They are called as anomers. Now, what is anomers? What is anomers? So there is a change in configuration at only one carbon, right? So this result into anomers. So when you write the anomeric structure, so at only the one carbon, you can see that there are two, okay, at one position, it will be, OH will be on the right hand side, on the another, you can see it on the left hand side. So they will give rise to anomer. So there is only configuration change at C1 carbon, okay. So this is called as so this will give rise to alpha D glucose structure and beta D pyranose structure, right? So they are called as D glucopyranose. So the two forms of D glucopyranose, they are called as the anomers. Okay. The vitamin that helps in clotting of blood is, so which is uh, the vitamin that occurs in clotting of blood, which is the vitamin clotting of blood, which helps in clotting of blood. It is K. Okay. So vitamin K, it is the vitamin which helps in clotting of blood. Yes, Karina, Amulya, and Lubna have answered it correct. Very good. Okay, let us go for the next one. So primary structure in a nucleic acid contains bases as GATGC. The chain which is complementary to the chain is. So you have to, so the primary structure is given in the nucleic acid. So you have to write the complementary basis for this. Yes. So GATGC is the primary base. Okay. So you have to write the complementary. We know that A is with T and G is with C. So right, tell me now, G will be C, A will be T, T will be A, G will be C, and C will be G. So it will be CTA, CG, option number C. Yes, baby has answered it correct. Understood. So these are the complementary. So wherever G is there, you have to write C. Wherever A is there, T is there, A. So these are the complementary bases. Yes, Lubna has also answered it correct. Very good. So primary structure, if they ask you in the nucleic acid, if it consists of AGATCG, then okay, this is DNA. Why? Because there is T, thymine is there. If there was GAUGC, that means it is for RNA. Okay. So this is the complementary chain. So option number C is the correct one. Next one, in alkaline medium, aniline exits predominantly as. So how does the alanine exits? How they exist as? So what is the structure of alanine? It is CH, COH, and NH2. So this is a structure of your alanine. So in alkaline medium, okay, what will be the pH? The pH will be high. So this will exhibit what? They will give the, so you will get NH2, CH2, and COO minus. So what it is forming? It is forming a Zwitter ion. Okay, it gives what? It gives a proton. So this will give proton, this will accept proton. So it will be in its Zwitter ion form. So it will be in its Zwitter ion form. Okay, so NH3 plus. Okay, so this will be in its Zwitter ion form. So option number B. Option number B. In alkaline medium, aniline will exhibit as the Zwitter ion. Okay. Next question. Alpha maltose consists of. Alpha maltose consists of. What does the structure of alpha maltose consist of? Yes. So if you see the alpha maltose structure, okay, it is made up of two glucose, right? So this is a structure HOH, HOH, OHH, then this is HOH, and this is CH2OH and H. Then here again HOH, this is HOH, OHH, this is HOH and CH2OH and H. So the linkage is present in here, right? So it will be like this. So what you are going to get? So this is a glycosidic linkage. So this is one. This is first carbon 1, 2, 3, 4, 5, and 6. Similarly, 1, 2, 3, 4, 5, and 6. Okay. So what you can see here? So alpha maltose, it consists of what? 2 alpha D glucopyranose structure. Okay. And what is the glycosidic linkage? The present at 1 and 4. So it consists of 2 alpha D glucopyranose with 1,4 glycosidic linkage. So it consists of 2 alpha D glucopyranose units with 1,4-glycosidic linkage. So option number D. Is it? Option number D is the correct answer. So yes, option number D is the correct answer. So see the structures. So structures are very important. Before coming to the conclusion, you need to know the structures. Next question. Sucrose is not readily reducing sugar since. Sucrose is not reducing sugar since. It is chemically stable. It contains no free aldehyde or keto group adjacent to a CHOH group. It is built up of fructose unit. It is optically active. Yes, sucrose is not reducing sugar. Why it is not a reducing sugar? So we know that all the sugars, okay, all the carbohydrates which contains the either the aldehyde group or the ketonic groups, okay, when they are in its hemiacetal or the hemiketal form, okay, they can reduce the tolerance or the Felling's reagent, okay, then they are called as what? Then they are called as the this what? Reducing sugars. Okay, now some disaccharides are there. Sucrose is what? It is a disaccharide. Okay, here what it consists of? It consists of two monosaccharide unit, and these two monosaccharides unit, they are what? They are having a reducing center. Okay, they are aldehydic or ketonic group. They are not reducing because the aldehydic or ketonic group, they are not reducing. So why? So it is a not reducing sugar. So why it is not reducing sugar? Because they do not have any free aldehydic or ketone group. Okay, because they do not have any free aldehyde or a ketone group. So because of that, it is not a reducing sugar. So option number B. Okay, option number B. So all the sugars, okay, all the sugars when they contains aldehyde and ketone group in their hemiacetal form or the hemiketal form, okay, they will reduce the tolerance reagent or the Fehling solution. Then they are called as reducing sugar. But here, when you see sucrose, one is a glucose, another is a fructose. So there is no free aldehyde or ketone group present. Okay, so because of that, it is not a reducing sugar. So option number B is the answer. Next one, dialysis can be used to separate. So dialysis can be used to separate A, glucose and fructose, B, protein and starch, C, glucose and protein, and D, glucose and NaCl. Yes, dialysis can be used to separate your glucose and protein. Okay, so option number C. So whenever dialysis is there, okay, you can separate the glucose and protein. Fine. Option number C is the answer. Glucose when reduced with HI and red phosphorus, what it gives? N-hexane, N-heptane, N-pentane, N-octane. This is the reaction used in elucidation of glucose structure. Yes, yes, tell me. What is the answer? It is N-hexane, right? So carbon, glucose structure, it consists of 6 carbon. Right. So when it gives, when you reduce it with HI, hydrogen iodide, in the presence of red phosphorus, what it gives you? N-hexane. It gives you N-hexane. Right. So option number A. Next one. The statement that is not correct. Which is not correct? Read out the aldose or ketose sugar in alkaline medium do not isomerize. B. Carbohydrates are optically active. C. Penta-acetate of glucose does not react with hydroxylamine. D. Lactose has glycosidic linkage between C4 of glucose and C1 of galactose unit. Yes, which is not correct. Which is the statement which is not correct? Yes. Yes. Aldoses or ketosis, okay, when they are in its alkaline medium, they do not isomerize. Okay, they do not isomerize. So this is not correct. Okay, they will, when they are in alkaline medium, they will isomerize. Okay, so option number A is the incorrect statement. Yes. Chelosis and digestive disorders are due to the deficiency of. So when you can see this deficiency, yes, when it is due to riboflavin. Okay, it is due to riboflavin. So when you observe riboflavin, so the deficiency of riboflavin, it will cause these disorders. Okay, chelosis and digestive disorders. So option number D. Next one. Adenosine is an example of what is adenosine? Is it a purine base, nucleoside, nucleotide, or pyrimidine base? What is adenosine? Yes. What is adenosine? Adenosine. What is adenosine? So whenever you see, yes, it is a nucleoside. Very good. What does nucleoside consist of? It consists of nucleic acid, okay, and a sugar. When nucleic acid is combined with sugar, okay, and a nitrogenous base, Okay, so nucleoside, what it consists of? Two components. It consists of nucleic acid, a pentose sugar, and a nitrogenous base. Okay, so in when you see the adenosine, okay, so what is adenosine? It is a nitrogenous base is what? Which is the nitrogenous base? It is adenine. So adenine. Okay, so this is the sugar. So it is a nucleoside. So option number B. Okay, option number B. Okay, last question. Which of the following gives positive test for Felling's solution? Sucrose, glucose, fats, and proteins. Which will give a positive test for Felling's solution? Yes, which should have a free aldehyde and it is a glucose, right? Glucose. So glucose, what it will do? Glucose, it will give you a Felling's test. Okay, it will give you a positive test for Felling's. Okay. So Felling's test, it is given by what? It is given by aldehydes. In sucrose, as we know, there is no free aldehyde group. Okay. In glucose, there is a free open structure. Okay. And the aldehyde is free. So yes, it is option number B. Is it clear? So these were some few questions based on the chapter biomolecules. Okay. So very simple chapter. Okay, to the point the questions will be. So yes, so these are some of the questions from the chapter biomolecules. Okay, is it clear? Any doubts? Okay, so this completes your chapter biomolecules. So in the next class we are going to take the next chapter. So 13th one, which is 13th one? Okay, in nucleic acid, the molecules are joined together by water bond. Okay. In nucleic acid, the molecules are joined together by phosphodiester linkage. Okay, option number B. Option number B is the answer. So now nucleic acids. Okay, what are the nucleic acid? In nucleic acid, there are the bases. Okay, A, T, G, and C. So to join these bases, okay, so there should be some linkage, right? So which is the linkage which is present? It is a phosphodiester linkage. So these phosphodiester linkage, what they do, they will join the molecules. Okay, they will join these molecules together in the nucleic acid. Okay, so that's it. So phosphodiester linkage. Now, how we have the glycosidic linkage? Okay, how it will join the two molecules or two units of glucose in the maltose. Okay, how we saw. So similarly, nucleic acid, how these molecules they are joined? They are joined by these phosphodiester linkage. Okay, so this phosphodiester linkage is nothing but phospho. Okay, there is a phosphoric acid group. So there is one phosphoric acid group. molecule is present, and there will be two hydroxyl group. Okay, two hydroxyl groups are present. Okay, so they what they will do, they will form two diester. Diester means they will form two ester groups. They will form two ester group, and when they will form two ester group, so when they will so it will have two hydroxyl. So phosphoric acid, when it reacts with two hydroxyl group, they will form two ester groups, and they will release two water molecules. So they will produce what? Phosphor diester linkage. So this phosphor diester linkage, it is used to join the molecules, okay, in the nucleic acid. That's it. Okay, 23. Which is the 23rd one? Okay. Cirrhosis and digestive disorder. Okay, so your digestive disorder, they are due to the deficiency of. So the answer was riboflavin, right? Riboflavin. This riboflavin, it is caused, why? It is caused due to the deficiency of vitamin B2. Okay, it is caused due to the deficiency of vitamin B2. So this is a deficiency. Okay, riboflavin. So digestive disorder means what? You will not be able to eat the food properly. Okay, it will digestion will not be properly in your body. So chewing is not proper. So that will affect your body. Okay. Then this. Yes, riboflavin. It is caused by vitamin B2, and these are the disorders which are seen. Okay. So this is all theoretical. Anything else? Uh, if you know what do you mean by chillosis, okay, chillosis means what will happen, there will be small cracks near your mouth, okay, the edges of your mouth, okay, and there some disorder will take place, there will be some cracks or some bacterial, this will cause, okay, when the deficiency of vitamin b2 will be there, so this disorders will cause. Tulosis means near your mouth corners some cracks will be there, okay, and some microorganism or bacteria will be formed there, okay. So these are due to the deficiency of riboflavin. So riboflavin is nothing but it is caused due to the when you have less amount of vitamin B2. Okay, is it clear now? Fine. Okay. Any other doubts? So these were the questions. Okay, if you have some more, if you have any book, or you can solve from previous questions. Okay, solve the questions from biomolecules. Okay. Okay then. So yes, so thank you. Okay, we will end this session here. We will meet in the next week. Thank you.